you've often said that if genome engineering is a pie, then gene editing is a slice of it, and CRISPR is a sliver of that slice.
Correct.
I love the visual. You're the storyteller, but I still need help. Can you help me define these terms? What is genome engineering, the pie? What is gene editing, the slice? And what is CRISPR?
So I'll start big. The big story is humans re-engineering life. Life is essentially a coded language. And we talk about genetics. It's not just genetics, but it's the way that we humans pass this code generation to generation. So that's the big story. And then, so how do we do it? And so you could say that just finding a partner, everybody's just finding a partner and having a child, or domesticating a plant. That is a form of engineering life. Over the last relatively small number of decades, two things have happened. One is we have developed the initially very rudimentary and now less rudimentary compared to what's coming in the future ability to edit genomes. I think your listeners are sophisticated people, so they understand that genome sits inside the nucleus, which is the egg yolk in the cell, and the cytoplasm is the egg white, and the genome stores mostly the inherited information that passes generation to generation. And so now new tools like CRISPR genome editing allow for tech scientists, technicians, others, now high school kids, to make very specific cuts. So you can use an RNA guide to find a specific place on the genome, and then a nuclease, which is basically molecular scissors, to make a cut. And then you can either cut something out and let the genome reconnect automatically, or you can put like a little snippet of DNA there, which will, in many cases, just fit into that space. Mm-hmm. Now there are new tools, base editing, gene edit, prime editing, which don't require a cut, but essentially you can change one letter to another. So it's much less aggressive. As exciting as all of this is, these are just the very, very early days of genome editing. But maybe 10 years ago, people thought, well, the story of biology is the story of genetics. But genetics is just one piece of biology, what we call systems biology or multi-omic biology. So there's the genome, the epigenome, because almost every cell in your body has the blueprint for the whole body, but not every cell is trying to make its own independent body. And that's because we have the epigenetics, which are basically kind of traffic cops turning on and off, saying, do this, make skin, don't make blood or
genes express
whatever.
themselves.
Yes, exactly. Yeah. But there's the metabolome, the proteome, all these omes inside of us. And then there's the envirome, the exposome, all these things around us. And all these systems are dynamic and interactive. So it's not just the story of genetics, it's the story of biology. And it's the story of humans using these various tools and capabilities, one of which is just knowledge of humans coming together and sharing ideas to increasingly understand complex, not just human biology, but all of biology, and then use that knowledge to be able to make manipulations. And maybe some of those manipulations are genome editing. Maybe some of that is epigenome editing. Maybe it's some is manipulating different parts of systems biology. There are lots of different ways that we can imagine. That's why, and then CRISPR is just one tool for gene editing. So like the big story is re-engineering life. CRISPR is one tool among many, many tools for doing that.